Hi, I'm Dr. Michael McKain, an assistant professor in the Department of Biological Sciences and the curator of the University of Alabama Herbarium. Research in my lab melds together studies of flowering plant biodiversity with the exploration of genome evolution. Members of my lab focus on investigating how polyploidy, or having more than 2 copies of a genome, impacts gene expression, which ultimately leads to phenotypic variation in different species. Projects in the McKain Lab include describing how the extra genome copies within an invasive species, namely Johnson grass, have varied as that species has invaded the United States since its introduction in the early 1800s. We also are doing sequencing of the Joshua tree genome so that we can use that genome to assist in identification of novel genetic variation across populations that will help to hopefully save the species as it's facing problems due to climatic variation. We also work on the origins of C4 photosynthesis using the species Steinchizma hyans, or gaping grass, which is a native here in the southeast, to understand how plants transition from traditional C3 to the more efficient C4 photosynthesis. Ultimately, this research has implications for food production and food security. Then there's a new project in the McKain Lab that is just kind of getting off the ground. It's a collaboration with Dr. Greg Starr here in the Department of Biological Sciences. And what this is looking at is how epigenetic changes, so changes on the DNA that are not at the DNA level, so these are like methylation pattern changes and stuff like that, have impacted gene expression in plants in the Arctic as a response to climate change. So the Arctic has seen massive warming in the last 10 to 20 years. So we're interested to see how plants are responding to that. We have a really cool collection of samples dating back to the 90s that have been collected like every 10 years, so we can actually trace this through time. In the McKain Lab, we do what I like to call a field to bench to computer approach. What that means is that members of the lab have opportunities to do field work where you go out and you collect specimens, bring those specimens back to the lab and isolate DNA or RNA from them and make sequencing libraries, and then to ultimately assemble and annotate and analyze either chloroplast genomes, whole nuclear genomes, or transcriptomes of those individuals. Currently, the McKain Lab has 5 undergraduate researchers and 4 graduate students, which this means there's a lot of opportunity for interacting with a community as you would grow as a researcher. We're always looking to add people, and we're excited about the opportunity to see people join the lab, grow as a researcher, and then ultimately contribute to publishable science. If you have any questions, feel free to email me. Thanks.